So I do this class in the afternoon right before this, and then do it again for you. And I actually have a little bit more time in the afternoon. That class is 3 hours and 20 minutes long, and of course 20 minutes break. You guys are just 3 hours long, still taking 20-minute break. And I was really rushed at the end of the last class, so I don't have great hopes for today. But I think part of what made me rush is I spent a little bit too much time at the beginning talking about some sort of basic housekeeping issues. So I do need to talk about a couple of things, but I'll try to say them more quickly. The first thing has to do with the fact that there was nothing there for you to look at before you came in today. I am going to have that be a regular part of the course, but that didn't happen this week because this weekend I went to a workshop, conference, whatever. And so I was kind of busy going to the workshops at the conference. So that took up a lot of the day on Friday and Saturday. However, I was out of town in a hotel, so after the workshops were over, I basically just got to sit around in my hotel and do stuff. Except that I was at a hotel and their Wi-Fi connection was a little spotty. So it took me a little longer to put the stuff together and get the A&P1 stuff ready. I do theirs before yours because I teach them on Tuesday and I teach you on Wednesday. And so I just didn't get around to the A&P2 materials, get it ready to post. So it's not there, which means that I'm going to today kind of default back to the more lecture-oriented presentation style, which we'll see how that goes. So hold on a sec. There we go. I forgot to tell the guys earlier about this, but also while I'm here, I want to mention a couple of things. First, I was going to say there would normally be, something here between the reading assignment and the part 1 assignment there. And what I'm going to do is a little bit different. So let me just jump to an A&P1 class where I've done this for them. So the sensory systems and autonomic system presentations that you had, there's just a link right to that presentation. What I'm going to start doing instead is there's now going to be a folder, and if you go into that, it'll have a number of presentations. Now, really what I'm doing is I'm taking sort of one big presentation and chopping it up into smaller bits. So for instance, this for A&P1 is about the cell biology part of the course. And so I've just broken it up into the main topics, and each one has sort of a smaller presentation. Cell membrane, organelles, nucleus, proteins, and cell cycle. I was already kind of thinking about this, but at the workshops, it kind of came up there a little bit more, that just the idea that it's better if I'm going to present this stuff to you at a time that it is broken up into smaller bits. If you go, oh my God, I got an hour and a half thing to watch, you're going to keep putting it off because you don't have an hour and a half to do it. Okay? But if you have a 10-minute thing to watch, or if you have 5 10-minute things to watch, then you might go, well, I can get one of them out of the way and then come back and do another one. Now, you could, if you had an hour and a half thing to watch 10 minutes of it and come back to it. But it's like not having commercial breaks in a television show. You're not really comfortable with where you want to stop, and it can just be daunting, so you don't do it. So I've kind of done that for you. I'll try to make them fairly short like that for you to follow. Short like that so you can deal with them in sort of bite-sized pieces. And if you need to go to one particular thing, go back to look at something, you know what topic it is within there. So it'll be like that. So you'll see that kind of thing there. And I'm going to do it for the blood. I definitely want to do it for the blood because I never have enough time to do the blood stuff with the lab that we're doing next week. So I need to give as much of that to you ahead of time as possible. Now, I do want to point out here in the endocrine system folder that there's something new. Now, if you had me for AMP1, this isn't exactly new. I've seen these before, but I want to point this out because it is different. It says here, overview assignment. This is just an announcement. You don't see the assignment here. It's sort of like the parts 2, 3, and 4. You don't see them until it's time for you to take it. But there are 3 points in the semester that you'll be taking these overview assignments, and they are meant to be assignments that encompass information from multiple chapters. So this one is going to be about the nervous and endocrine systems, sort of comparing and thinking about what they do. And you'll do part 1. When you have that done, you go into part 2. You have that done, you go into part 3. You have that done, you go into part 4. For the overview assignment, you just have to complete part 4 to go on to the overview assignment. Just complete that one and the link will appear. Now, it'll be different from the other ones. The other ones you have to get 80% correct to go on to the next one. But you have to get an 80% score on it to go on the next one. But for the overview assignment, you just have to finish part 4. Now, those are manually graded. I can't have you going back in and redoing them over and over again until you get a certain score. So that one's not score dependent to go on. If you do that one late, it won't hang you up to go on to the overview assignment because it's not dependent on the score. So like if you do part 2 late, you can't go into part 3 until I check it off because it holds up the score until I check it off. But since doing the overview assignment is just based on completing part 4, then once it's done, you'll be able to do the overview assignment. Even if it's late and I have to check off and grade that thing. It'll work out fine like that. You'll also have one with the cardiovascular system to tie together those 3 chapters. And you'll have one with the environmental exchange section of the book, which is respiratory, digestive, and urinary systems, because they're all intertwined. There's a lot going on between them. So you'll have those sorts of assignments there, and for all of them, you'll see a little blurb saying that it's there because you don't actually see the assignment. But then when you've completed part 4 for that chapter, you'll see another link show up here, which is that assignment. So you know what you have to do it. Okay. So yeah, that's all I wanted to say about that. So I want to go into the presentation to explain the endocrine system. I will try to have this up and available for you like the other chapters, but it'll be broken up into smaller bits. But I hope to have that done by the weekend, but I have this big meeting on Friday that's going to really screw up my schedule anyways. So I want to be prepared against not getting it up in time for you to complete the assignment. So I want to sort of do a little presentation here for this. Now I'm going to break, like I said, I'm going to break it up into different parts. And so for each part, it'll have, endocrine system because it's the unit, and then the title of that part. So I'm going to start with an overview of it. Then I'm going to have a thing about the hormones and the hypothalamus and the pancreas and growth hormone. It'll just break it up into several parts. There are a few pictures in here. Actually, I think there's only one that I need to change out. One of the things I'm trying to do with these presentations, one is to be sure that all the artwork that's in the book is represented here, so I can sort of explain to you what the book's covering. But also in places where the book doesn't have artwork that I want to use, I want to add artwork to it. But I want to use stuff that's not copyright infringement. So I actually do have a picture in here that's copyrighted because it's from an old book, and I haven't gotten in to replace that. So I'll point that out, and when I do finally post it, I'll have a copyright-free version of that information in there. So, now, just as an overview of the endocrine system, when textbooks talk about the endocrine system, they usually list these 10 glands as being part of it. Actually, there's only 9 represented here, but the 10th one's in the picture. So I just want to quickly run through them and mention what they are. I'm going to start at the most superior and then move down to the most inferior. So the first one is the pineal gland, which is this little gland that's part of the brain. It's actually part of the thalamus, part of the diencephalon, which means technically it should have the word thalamus in its name. And the other name for the pineal gland is the epithalamus. Not that that's terribly important, except that in A&P 1, I point that out whenever I explain the structure of the central nervous system. It's called the pineal gland because it looks like a pine cone. That's just where it got the name. It only releases one hormone, which is melatonin. Melatonin is a signal to your body about when it's nighttime. During the day, when sunlight is visible, whether it's directly visible like right now and the sun shining down, or there's clouds out and it's filtered through that. Sunlight has a particular color to it. Actually, it has all colors to it. It's white light. And our retinas are sensitive to color vision. And so our retinas know when we're looking at sunlight as opposed to artificial light, like what's in the the ceiling here. And so that color information about the sunlight comes in through the retina. It actually goes to the hypothalamus. And then the hypothalamus tells the pineal gland basically if it's night or day out. And when it's night, the pineal gland releases melatonin. Melatonin released from the pineal gland sets up our internal clock, our diurnal or circadian rhythms. Without input from sunlight, it would run at a slightly longer than 24-hour period, 26 hours or something like that, I can't remember. And so it's sort of reset every day with the sunlight so that we keep in sync with the actual length of the day on outside. You can fake it out by exposing yourself to white light. And some people have mood disorders, especially in the winter, seasonal affective disorder. Because there's less sunlight, it can actually cause a depression, series of symptoms. And you can fix that by faking out your brain by looking at white lights. My mother-in-law actually has a little bit of an issue with this herself, so she owns what's called an OT light. I don't know why it's called not light, must be the brand name. But it's a special light bulb that's brighter and puts out more energy so that it's a broad spectrum light, as opposed to a fluorescent bulb, which is actually a little bit on the green side, or an incandescent bulb, you know, the regular light bulb looking shape thing. Those are a little bit on the yellow side because of the temperature that the filament burns at. So you can fake out your brain by exposing yourself to broad spectrum light. And so somebody with seasonal affective disorder turns that light on as the sun goes down and makes the brain think that there's still sunlight out there. Helps them regulate mood a little bit. Then the next thing isn't actually in the picture here, although it's right here. It's the hypothalamus. So there's a bracket that says thalamus, which is essentially where the thalamus is right here. So below the thalamus right there is the hypothalamus, kind of where these little pointy things are at the corner of that bracket. That's where the hypothalamus is. There's a better picture of it in a image I'll show you in a little while. That's color-coded showing you where it is, but that's where it is. It's just superior to the pituitary gland, which is an important point to make. It's the master control center for the endocrine system, which is why it's kind of odd that it's not actually pointed out in this picture. But now it's the master control center really for homeostasis, and it controls the endocrine system, which we're going to talk about tonight. It also has some effects controlling the autonomic system, which we didn't really talk about last week with last week's material, but it is true. For a number of autonomic functions, the hypothalamus can regulate them and also coordinate it with higher mental functions. So it does happen. We just didn't stress that last time. The hypothalamus releases two hormones directly into the bloodstream. Well, actually it releases a bunch of hormones in the bloodstream, but two of them travel throughout the body and have targets quite a ways away. And the rest get in the bloodstream and have targets directly in the pituitary gland. Okay, so they don't really go very far. And a lot of what the hypothalamus does to control homeostasis through the endocrine system is by controlling the pituitary gland, which brings us to the pituitary gland. There's actually two halves to the pituitary gland, the posterior and anterior parts. The posterior part is really just an extension of the hypothalamus, and it's where those two specific hormones are released. They're called oxytocin and ADH. Oxytocin is related to reproductive functions. Most importantly, it causes contractions of the uterus during childbirth. ADH is responsible for controlling water balance in the body. ADH stands for antidiuretic hormone. Diuresis means to pass water or urinate. Antidiuresis, therefore, means to retain water. And so this hormone causes you to retain water instead of passing it in your urine. Just a quick overview there. The anterior pituitary is told what to do largely by the hypothalamus. The hypothalamus has a number of hormones that control that. But then the anterior pituitary makes its own hormones in response to that and releases those into the body. And a lot of those will actually tell some of these other glands what to do. So then we move down, we're in the throat region, and we have the thyroid, which is sitting right at the base of the larynx, the voice box. And so here's the larynx right here, and the thyroid's wrapped around it. This is actually a posterior view, which is a little bit misleading. On the posterior side, the two lobes of the thyroid gland don't actually meet in the middle, but they might come kind of close. Okay. But the reason it's showing you the posterior side is because on the posterior side of the thyroid gland are little glands called the parathyroid glands. They vary in number from person to person. Someone might have 4, like this picture suggests. Someone might have 8. The number is not really important. It's really about the total amount of tissue. So 4 big or 8 little glands are going to have the same sort of effect. And I'm going to talk about the function of those a little bit later. Then we have the thymus, which sits on top of the trachea and on top of at least the superior part of the heart, which is not in this picture. When we look at anatomical models, we don't see the thymus because it blocks our view of the heart. So like the torso models that we have in lab, the thymus isn't there because it's in the way of seeing the heart. But also it's not there because in the adult, which is what those models are representing, the thymus isn't active anymore. It's only active during childhood and adolescence. And by the time somebody's an adult, it's really just fatty tissue and doesn't do anything. Okay. And then in the picture, we have a big gap from the thymus down. The next one is the adrenal glands. Adrenal means on top of the kidney. You may find sources that refer to this as the suprarenal gland, which means basically the same thing, just that apparently there are some people that are trying to change the name of the gland to reflect the idea of superior on top of. But really, that's what ad means. So personally, I'm in the adrenal gland camp of naming things. Then we have the pancreas, which lies just posterior to the stomach and a little bit inferior to it. That's a big player in understanding endocrine function. We'll talk about that a little bit later. But it's really more a digestive gland than an endocrine gland. So, but it does have endocrine function. And then we go down from that and we have some reproductive organs. The ovaries and the testes are the reproductive glands, making the gonadosteroids, I guess you'd call them, the gonad hormones, steroid hormones like testosterone from the testes or the estrogens from the ovaries or progesterone. Estrogen is not a specific hormone. It's a group of hormones. The most common one is estradiol, but they're often referred to collectively as the estrogens. And in popular usage, a lot of people will just treat it as if that's the name for the hormone. It's not the hormone. Testosterone is a hormone. It belongs to the group called androgens. So estrogen and androgen are equivalent terms. Testosterone and estradiol are equivalent. Okay. And it also points out the uterus, which releases hormones that help to regulate various functions during gestation of a fetus. Generally speaking, those 10 things, well, 11, including the uterus that I just mentioned, but those 10 glands are usually thought of as the main endocrine glands. I don't know why those 10 are necessarily the ones that are always mentioned, because there are more things that have endocrine function, and these are not necessarily strictly endocrine glands. Some of them are, but not all of them. And so, like, the big gaps that we see here between the thymus and the adrenal glands, we're jumping over, like, the heart's in there. The heart releases hormones also, which helps to regulate blood pressure. They're not labeled here, but the kidneys are obviously there. The kidneys are important too. They also contribute to blood pressure regulation, but they're not considered endocrine glands the same way these others are, just because they didn't make the cut on somebody's book. We're not going to talk about all of these. We're only going to concentrate on a few of them. Some things and some of the things that aren't listed here will come up as we're talking about those organ systems. So when we're talking about, the kidneys, we'll talk about the heart and the kidneys' role in blood pressure. When we get to the digestive system, we'll see that the stomach and the intestines release a good number of hormones that have important effects. When we get to the reproductive system, we'll talk about those structures there. In the endocrine system, the immune system, we'll talk about the thymus when we get to that. So really today, I've said everything I want to say about the pineal gland because it's not that exciting. So today we're going to look at the hypothalamus, pituitary, the thyroid, parathyroids, and the adrenals and the pancreas. Okay, that's what we're going to concentrate on today. But before we get to that, I want to talk about hormones. Before I get to hormones, I just want to point out up here at the top, these are sort of the big functions of the endocrine system. To some way we could, or just in one way of thinking, these are kind of overlapping things. Regulation just means, kind of the homeostatic mechanisms. And growth and development is homeostatic in a sense, not the same way as like keeping heart rate normal or whatever, but there's feedback loops and certain ways that things are supposed to progress. And all of that kind of depends on the hormones input. Now, you can probably tell that there's some stuff written underneath that, those two words that you can't read at this distance. You're welcome to zoom in and look at those on your own. It just sort of defines more specifically. But I'm going to move on to this picture down here. Okay. So I have this picture. It's out of the cell section of the book. Coincidentally, I was just showing you that part of the MP1 course talking about the presentations. But the reason I have this here is because it represents the secretory pathway, which is important for the endocrine system. Not all hormones do exactly what I'm going to talk about here, but it's essentially this, or they make use of parts of this. So the nucleus has the DNA in it, and DNA is instructions for making proteins. Okay? And many of the hormones that we're interested in are protein hormones, which we'll learn about in a little bit. The DNA is transcribed into RNA, and then the RNA moves out into the cytoplasm where it is translated into proteins. And for the secretory pathway, that always happens in the rough ER. Okay? Ribosomes attached to the endoplasmic reticulum translate the mRNA into a protein, and then that protein travels through the maze-like structure of the ER and is eventually released within a transport vesicle, which will transport it from the rough ER to the Golgi apparatus, and it'll move across the Golgi apparatus, and it'll be processed and eventually get packaged into another vesicle that's going to secrete that out into the extracellular space. Okay. Now, again, this is just out of chapter 3, and it's not the endocrine system chapter. So what they're depicting here isn't actually a hormone. This is just a membrane protein here. So it's in a vesicle, but it's sort of sticking out of the wall of the vesicle. And then when the vesicle merges, it becomes part of the membrane. But really, the hormones would be in the vesicle, and they'd be released into the extracellular space. We're going to talk about insulin a little bit more later on, and I just want to use insulin as an example here. So there's a gene for insulin. It's transcribed into mRNA, and then mRNA is translated into a protein that travels through the rough ER, and then is transported onto the Golgi apparatus, or into the Golgi apparatus. In the Golgi apparatus, the insulin molecule is modified. What we actually secrete as insulin is a little bit different from what's originally made. I don't quite remember these numbers, so I might be kind of making this up, but let's pretend I know what I'm talking about. The insulin protein, or the insulin gene, codes for 156 amino acid long chain. I might be way off the number, but trust me, it'll make sense in a second. So that chain would be just a straight chain of amino acids, but what happens is, actually, I guess this first thing happens in the rough ER. It folds up into its final shape, and a two-dimensional representation of part of that shape is just going to be that the thing folds around into a loop. So it ends up looking like this. Okay. There's amino acid residues in both chains, or both sides of the loop, that will link together. They'll form covalent bonds and hold it together in that loop shape. Okay. It has a bit more three-dimensional shape than this, but that's the basic idea. Okay. Then in the Golgi apparatus, what will happen is the protein will be cut right there and right there. Okay. And so now what did I say, 156? Which now that I say this, no, let's say it's 156, is now 15 amino acids are removed from it. I think that's right. And so we have left over 141 amino acids that make up actual insulin. What's cut off here, this part is called the C protein. Okay. It's called that because the other two parts are A and B. Okay. So this is A, this is B, this is C. But C is actually removed from the molecule, And active insulin is really just this part here. Of course, it has a bit more three-dimensionality to it, but that's the idea. What's released at the end, because that C protein's cut off while it goes through the Golgi apparatus, it's still with insulin when it's in the secretory vesicle. And so what's released at the end is insulin and C protein. If you ever look into more detail about insulin, or if you ever work with patients that have diabetes, you might see reference to C protein, maybe they call it C peptide, I can't remember, levels. What it is it doesn't have a whole lot to do with insulin's physiological effects, but every time a cell secretes insulin, it's secreting the C peptide also, or C protein also. So it's a very good marker to measure the level of insulin without interfering with insulin doing its job. And there are different types of diabetes. If the diabetes is because cells are not secreting insulin properly, then C protein levels will be low. But if the diabetes is because of some other reason, C protein levels will be normal, and the insulin's is failing somewhere else in the pathway. So it's from the secretory pathway, you can kind of learn a little bit from that. Yeah.
What's a C-reactive protein? I've seen that on a blood test. Is that similar? Or?
Is that in regards to diabetes?
I think it's a inflammation marker.
Okay, yeah, then that's what I thought. It's a totally different story. People naming molecules aren't usually that inventive, so there will be a lot of things named A, B, or C, or D. So it's a coincidence they're both C something. But, I'm not entirely sure what that is. I've heard it before. I just don't know what it is exactly. Okay. This picture is actually copyrighted, and I will update this later on to fix that. If you're watching this recorded thing on YouTube right now, please close your eyes and don't look at this because I'm infringing on copyright. There's supposed to be a lot of laughter after that part, but whatever. Now, this is a picture out of an old book, and it addresses something that our current book addresses too. There's just not a picture in the current book. This falls a little short. I need to add a little bit to it, and actually, I can't find a suitable picture on the internet to replace this, so I'm going to have to draw my own. So when I draw my own, I'll be able to make it show exactly what I want to show. This is showing different types of communication. Now, really, as far as this course is concerned, there's two types of communication. There's nervous system communication, there's endocrine system communication. Okay. What this is getting at is that there's a little bit finer detail within that. Okay. Now, synaptic communication is what we see in the nervous system. Okay. At the end of an axon, there are vesicles that release neurotransmitters across the synapse to bind with the receptors on the target. Okay. And the distance between where they're released and where they bind is very, very short, measured in micrometers or nanometers. Okay. Now, these are secretory vesicles, just like what I was just showing you, except that they're nowhere near the Golgi apparatus, and they're usually packaged here at the end of the axon. That's just the way that things work here. Because the axon is what covers the distance between the neuron cell body and the target. Okay. Now, endocrine signaling is what we're talking about tonight, and we usually characterize it as the signaling molecule, called a hormone, is released from the cell, and then it travels through the bloodstream to the target. Okay. So in the nervous system, the axon goes out to the target. In the endocrine system, the hormone travels through the bloodstream to get to the target. But it's a little bit more complex than that, because the cells that are releasing the hormones actually have no real control over where the hormone goes. And there's actually 3 different things that hormones can do. One of them is the other picture here, which is called paracrine communication. Now, in the book that I got this from, I'm not crazy about the fact that they use a different word for what the chemical's called here as opposed to here. It's really still a hormone. And to some degree, calling something a hormone is a little bit artificial, but it can be the exact same molecule. This cell right here can release molecules into the bloodstream and can release molecules into that local tissue. And in the stomach, that very thing happens. There are cells that release hormones that have effects right there at that part of the stomach and travel through the bloodstream to affect other tissues elsewhere. Okay, so it's the same cell doing both things. Endocrine refers to traveling through the bloodstream. Paracrine means that it's affecting local tissue. Okay, so the hormones are traveling through the interstitial fluid rather than the plasma. Then there's a third one that should be in here just to complete the picture, but for whatever reason, this book didn't add it in. The book we're using does refer to all three of these things. Okay? Endocrine, paracrine, and the third one is autocrine. What does autocrine mean? Right, activates itself. So endocrine is kind of long-range activation. Paracrine is short-range activation. And so autocrine is the only thing that's left, which is self-activation. So this cell could be releasing hormones that will affect this neighboring cell, or it could just bind to a receptor on that cell right there. And the stomach is a great example of this. I mentioned the stomach is a good example for just these two, but autocrine is present in the stomach also. The stomach releases a hormone called gastrin. Very uninventive, just means stomach hormone. It's released from the gastric glands that make the stomach's digestive juice, the acid and enzymes that break down food in your stomach. Gastrin will cause gastric glands to be more active. So when your stomach has a lot of food in it and has to digest that food, gastrin levels increase to increase the activity of the stomach breaking down that food. So gastrin is being released from these glands. It activates itself to make more gastrin. It activates its neighboring cells to increase acid and enzyme production. And it gets into the bloodstream and can tell gastric glands and other parts of the stomach to be active too. So it does all three together. The cell itself doesn't have any control over where the hormone goes. It can fold right back and affect that cell that's releasing it. It can affect a neighboring cell, or it can get in the bloodstream and have more far-reaching effects. Okay. So it can do all of those. So autocrine, paracrine, and endocrine are really all things that hormones can do, as opposed to what neurotransmitters do at synapses, which is a very directed signal, because the axon is going out and reaching the target directly. So, okay, so I hope to have my own kind of version of this made up to include autocrine and sort of show how those three all can kind of work together. And also so that I'm not stealing a copyrighted piece of artwork. Now, this is not copyrighted. This is a picture out of the book. And the pictures I'm going to deal with from here on out are either out of the book or not copyrighted. What I want to talk about here is just the idea of what molecules are hormones. The book goes about this in one way to explain what the hormones are, and then shifts gears a little bit to talk about what those hormones do. I'm going to explain that, but I also want to set up a little bit of what the hormones are in terms of what they do, because I think that's an important thing to talk about. That'll be clearer when I get into explaining. So in this table, they have listed 4 different types of molecules that are hormones. Okay. I think I can zoom in on this. So the first type is called an amine hormone, which means an amino acid has been modified to make that hormone. Now, given the choice, I would not have picked norepinephrine as the example there, but that's the one that picked. But the amino acid tyrosine is modified into norepinephrine. Now, tyrosine is actually used to make a bunch of different stuff. Tyrosine is made to make dopamine, which is then modified into epinephrine, which is then modified into norepinephrine. So there are other things I could have put up here besides norepinephrine. I would prefer epinephrine as an example because it's more clearly a hormone. Norepinephrine is also used as a neurotransmitter in some situations, so it kind of muddies the water. But it's just an amino acid that's been modified. We can still see the amino group on it right here. The part that made it an acid has been removed. So it's an amino or amine hormone. There are a few other ones. Melatonin is actually derived from tryptophan. The thyroid hormones T3 and T4 are also derived from tyrosine, but they don't have all this other stuff on them. They're just the ring structure from tyrosine, pretty much. Those are the only ones that occur to me right offhand, but there might be a couple of others. Then we have peptide hormones, which are chains of amino acids. Okay? Now, what do we usually think of as a chain of amino acids? Protein, right. Which is the next thing here. Now, peptide hormones and protein hormones are basically the same concept. They're chains of amino acids. The difference in them is really just their length. Okay? So a peptide hormone is a short chain, and a protein hormone is a long chain. And sometimes protein hormones can be multiple chains that are together, which I can't tell from this picture if that's true of this one. But insulin is a protein hormone. Okay? In its final form, because the C protein's cut out, it is in fact two chains that are linked together. But because of that C protein thing, it's a weird little story for that one. This is growth hormone. I don't exactly know what it looks like in its final configuration outside of this little cartoon here. But these cartoons are kind of hard to read. So. The peptide hormone that we have here is oxytocin. It's 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids. I counted them in that order because the actual chain goes glycine and then loops around here. These two here, which are cysteines, there's a line between them because cysteine amino acids will tend to bond to each other in what's called a disulfide bond. So that kind of makes a loop there. And in fact, the lines that I've drawn in here holding the A and B parts of insulin together, those are disulfide bonds. So there's two cysteines there and two cysteines there that hook up to hold the loops together. And so that's what's happening. But this is just 9 amino acids. Now, I'm not an expert in biochemistry, but if I remember correctly, a peptide hormone like oxytocin or some of the other ones are usually protein fragments, which is not really a formal term here. But what I mean by that is that there's a gene that makes a really long amino acid chain. And then peptide hormones are made by cutting that chain up into smaller bits. Not positive that that's the way that oxytocin is made, but I wouldn't be surprised. But I do know that there is a whole class of hormones that are made from one big gene that are all cut up into little pieces. And those are really short, like 3 or 4 amino acids long. So, but those are peptide hormones, short chains. Proteins are long chains. And then at the bottom here we have steroid hormones, which are derived from cholesterol. If you look at a cholesterol molecule, it has this ring structure, these 3 6-sided rings and this 5-sided ring here, always configured like that together. When we have steroid hormones in our body, we have them because we've gotten cholesterol in our diet and modified the shape of the cholesterol molecule to be that. Okay. Now, cholesterol, just as an example, out here at this corner has a single bond to an oxygen, which is then bound to a hydrogen. Okay. So one of the things that's done to change cholesterol into testosterone is that hydrogen is taken off and the oxygen is double bonded back to the ring, as an example. Another thing that's different is Cholesterol has this like 5 or 7 carbon chain hanging off right here. That's obviously been removed from both of these. So there's some modifications there. But any steroid hormone is made from cholesterol. Okay. What type of macromolecule is cholesterol? Yeah, it's a lipid. Okay. And so steroid hormones are lipids also, okay, which is to say that they're very hydrophobic. Now, they might have some hydrophilic things on them, like OHs or a double bonded O or something like that. But overall, they're basically lipids. Okay. That sets up the actual, well, not the actual, but the other way of thinking about hormones, which is to classify them based on how they dissolve. So we have lipid-soluble hormones and water-soluble hormones. For what we have on the screen here, obviously steroid hormones are lipid-soluble because they are lipids. And then the protein hormones and the peptide hormones and the amine hormone that we have pictured here are all water-soluble because they have polar aspects to them. Proteins usually are water-soluble, which peptides would be too. And then amine hormones, it depends on what's there. Okay, so this OH and this OH and this OH and this NH2, those are all polar things, so that this will dissolve in water. Now, there are amine hormones that are lipid soluble, and those are the thyroid hormones T3 and T4. So when making T3 and T4, what happens is tyrosine, which has stuff like this on it, all of that gets removed. Maybe one of these things stays. And just that ring structure is used to make the hormone, which is pretty much a lipid. It's hydrocarbons. Actually, 2 rings are used. So the thyroid hormone is often depicted something like that, representing the 2 ring things. There's more to it than just that, but that's sometimes a cartoon representation of hormone. I used to have pictures that had thyroid hormone depicted that way. I don't use them anymore because they're copyrighted, but you might see that in other sources, just depicting a lipid-soluble form of an amine hormone because it's really only that part of the amino acid that's not formed. So it's really a matter of whether they're polar or non-polar molecules if they're going to dissolve in water. And this kind of defines that. So we have lipid-soluble hormones, which are based on hydrocarbon molecules, meaning lipids. And the best example of that are the steroid hormones. And we have water-soluble hormones, which are just polar. So they might be fairly unmodified amino acids, meaning that they're still polar, or they might be chains of amino acids like peptide and protein hormones. There are a type of water-soluble hormone that's actually derived from hydrocarbons. They're called, well, as a group, they're called eicosanoids. Don't worry about that name. The most common type of that is prostaglandin. Has anybody ever heard of prostaglandin? What context have you heard prostaglandin in? Prostaglandins are made from lipids, and the enzyme that makes them is called cyclooxygenase, which is abbreviated COX. Okay, so it's the COX enzyme. You are probably familiar with prostaglandins, maybe not so much by name and definitely not by enzyme, because people take a type of drug called a COX inhibitor quite a bit. Okay? There are actually 2 COX enzymes that are important, COX-1 and COX-2. And a drug that's taken very commonly is a non-specific COX inhibitor. It just blocks prostaglandin production. Okay. Before I give the big reveal, does anybody know what the common COX inhibitor drug is?
Acetaminophen?
You picked the exact wrong example.
Okay.
It's aspirin. So acetaminophen or Tylenol works similarly to analgesics like aspirin, but It's not a COX inhibitor. So, but aspirin or ibuprofen or naproxen or that class of NSAIDs are COX inhibitors, and they block the production of prostaglandins. COX-2, when it makes prostaglandins, is making prostaglandins that are part of the inflammatory response that cause pain, and that's why people take aspirin for pain. However, there's a side effect of aspirin and NSAIDs. What is that? No, not quite that severe. What? Stomach, yeah. It can cause ulcers and stomach problems because COX-1 that makes prostaglandins is active in the stomach lining because prostaglandins act as a buffer against stomach acid. So if you take aspirin, you're going to block COX-1 in the stomach, which means that you won't buffer your stomach against stomach acid very well, which can lead to stomach ulcers. And that's why it's a side effect of that. Okay, so I'm trying to cut down on how much I talk. So I'm not going to go on with that story. I just wanted to connect it to something you've heard of. But I could go on for another 10 minutes about COX inhibitors. But that's an example of a hydrocarbon that has hydrophilic functional groups. That's what happened to make prostaglandins. They're water soluble. Okay. Now, the reason why solubility is important is because hormones have to be transported. They move through the bloodstream. What is the blood chiefly made of?
Water.
Water, right. And so this becomes an issue for what the different types of hormones can do. Now, water-soluble hormones can obviously dissolve in plasma easily, so that's not a problem. Where it's a problem is lipid-soluble hormones. They don't want to dissolve in water. So they need to have a specialized transport protein to help them get through the blood, for them to sort of sneak inside of that protein, and the protein dissolves in the plasma for them. The thyroid hormones, T3 and T4, get special transport proteins made by the thyroid gland to help them get through the blood. Steroid hormones take advantage of a protein that's already part of plasma called albumin, and just slip into little pockets there to be protected from the water. And next week we're going to look at blood, and that'll come up when we're talking about blood and plasma proteins. So transport's an important issue there, but then the other issue has to do with how these hormones interact with their receptors. Now, first off, a word about receptors. Hormones only have an effect when there's a receptor on the target cell. Okay, I could take growth hormone and dump it on your head and you wouldn't grow, okay? Because the cells on top of your head do not have growth hormone receptors. But if I apply growth hormone to the liver, that's going to stimulate growth because of how growth hormone works. And I'll talk about that before the end of the day. But now, actually, none of you will grow because you're done growing. But for somebody that still has growth plates in their bones, applying growth hormone to cells that have growth hormone receptors will, in fact, cause growth. But if you just put growth hormone on a tissue that doesn't have, or a cell that doesn't have any receptors, it has no effect. Receptors are necessary to make a hormone have an effect. Okay. A target cell is defined by having a receptor for that hormone. Now, lipid-soluble hormones, because they're lipid-soluble, can cross through the lipid bilayer of the cell membrane. Water-soluble hormones cannot. Okay. Now, water-soluble molecules might be able to cross the cell membrane if they have a channel or a transport protein to help them do that, but that's not the case here. Instead, water-soluble hormones deal with receptors that are on the surface of the cell. They don't get into the cell. They tell the cell that they're in the area, and then the cell reacts to that. Whereas lipid-soluble ones go into the cell and do their job. So these are two pictures from the book. sort of illustrating the difference between these. So we have a cartoon describing what happens with lipid-soluble hormones, and then one for water-soluble hormones. Let me just zoom in on this one a little bit. So there is a little problem with this picture, which I'm not going to get too hung up about, but I do want you to realize that it's present. The lipid-soluble hormone that's pointed out up here in the capillary would not be floating around by itself in the bloodstream. It would be tucked into a transport protein. When it releases from that transport protein, it'll move out of the blood and into the extracellular fluid, and it'll head right for the cell, where it will just move straight through the cell membrane and get inside the cell. And then inside the cell, it binds to its receptor. Then the hormone receptor or receptor hormone complex acts as a single unit, which then has an effect in the cell. Now, this is a cartoon, and it's not meant to be entirely perfectly accurate. Some sources will say that receptor hormone complexes form in the nucleus, which is probably most common, especially for steroid hormones. But it's possible that receptor hormone complexes form in the cytoplasm, and is probably more common for the thyroid hormones. So where exactly it happens isn't crucial. What's important is the receptor hormone complex will then have an effect, usually in the nucleus. And as a complex, it binds to the DNA. It will recognize a specific sequence of DNA bases and bind there to recognize a particular gene. And by binding there, it'll probably increase the activity of transcription to make mRNA of that gene, which can then be translated into new proteins. Now, the best example of this idea is in anabolic steroids. Who can tell me what an anabolic steroid is?
Steroid that makes your muscles bigger.
Okay, a steroid that causes tissue growth. And the most common examples of those are ones that cause muscle mass increase. Why are you familiar with anabolic steroids?
Big.
Arnold Schwarzenegger.
Because they're used as performance-enhancing drugs, often by athletes, whether it's in baseball or weightlifting or whatever. And all that's happening there is by injecting testosterone or a testosterone analog, it'll get into muscle cells and simulate the production of those proteins that make up the myofibril: myosin, actin, titin, the Z-disc proteins, those sorts of things. Make more of those proteins, you make more myofibrils, and therefore the muscle can generate more force when it contracts, okay? And there will be more mass, it'll be a bigger muscle. Okay? Probably, and I'm not positive about this, but probably it also stimulates the growth of new myoblasts to make more muscle fibers overall, but definitely it'll cause muscle protein transcription. Thyroid hormones can do similar things, increasing the production of enzymes that help catabolize glucose, as well as getting into the mitochondria, where it'll also increase mitochondrial activity to make more ATP. Okay? And also something I read once, and I just didn't follow up on much, but it makes sense. It'll also increase activity in certain cells, like cardiac muscle cells will beat faster. They've got a bunch of energy because the mitochondria are making more ATP, so why not use that ATP? So thyroid hormones do that kind of thing too. This other picture here shows the way that receptors work for water-soluble hormones. Now, in this picture, the water-soluble hormone up here in the capillary, it's completely accurate to represent it as floating around by itself, because that's what they're going to do. Now, if I were the art director telling somebody what to do with this picture, I would have said, hey, doesn't that look like a pimple? let's not draw that in the picture, but that's how they've represented it. That's not what they actually look like. The receptor is a protein that spans across the cell membrane. It's not a little, puckering out of the cell membrane, but it has on the outside surface of the cell a place that the receptor of the hormone will bind. And then on the inside of the receptor, this thing that looks kind of like a bow tie for our pimple, is a protein called a G protein. It's an enzyme. It hydrolyzes a molecule called GTP, which probably sounds familiar because you've heard of ATP plenty. GTP is just a similar energy-rich molecule. And by hydrolyzing that, it gets energy out of GTP, which allows it to physically move from the receptor protein over to another protein here, which is more generally referred to as the effector protein. In the book here, in a lot of explanations of this that you might find, they're going to give you this specific example because the majority of hormones work the way that this picture is showing, using this enzyme called adenylyl cyclase. When the G protein binds to adenylyl cyclase, the effector protein has its effect. What it does is it hydrolyzes ATP, and it actually removes 2 phosphates, getting down to AMP, or the monophosphate version of it. The cyclase in the name of this enzyme is doing something a little quirky, which isn't terribly important other than it tells you about its name. The lowercase c here is for cyclic. So this is cyclic AMP, which means that in this molecule, the enzyme cyclase forms a bond between 2 parts of the molecule to make it circular in structure. Okay. The biochemical nature of that is not important. It just happens to be the case. And I'm saying it so that you understand why there's a little C there. CAMP is different from just plain old AMP. Okay. Now, this molecule here is what's referred to as the second messenger. In the book, they call it the secondary messenger, which bothers me, but is probably grammatically more correct. But in most every other source, you'll hear it referred to as the second messenger. If this molecule is the second messenger, what's the first messenger? Nope, a little further back. A little further back. The hormone itself, right. The hormone can't get into the cell. It's the first message that arrives at the receptor. Binding to the receptor causes the second messenger to be made. Second messenger is only there because the first messenger showed up. Okay. Because the hormone can't get in, it has to cause these metabolic changes to create a signal inside the cell. Okay. Now, there are other systems that use other things besides CAMP as a second messenger, but by far the most common one is this, and that's why that's exactly what this picture is showing. The second messenger then can go on and cause changes. It can activate certain types of enzymes. It can activate other types of proteins. It can cause metabolic changes in the cell. What's not pictured here, but it's also possible that a second messenger could cause gene transcription in the cell. Okay. So there's a lot of things it can do, but whatever it does, this only shows up, the second messenger only shows up because the hormone arrived in the first place. Okay. Now, this picture from the book is not great at explaining or showing the parts of the receptor complex. So I actually want to move real quick, and I meant to actually put this picture in the presentation while I was eating dinner, and I just forgot. So this is a little schematic representation of the receptor complex here. This is called, and I don't know if anybody happened to see it written up on the board earlier, I erased it. What we're looking at here is called a G protein coupled receptor. which is abbreviated GPCR, if you come across that. It has three components. First is the receptor protein. And that's what the hormone actually binds to. And then second is the G protein, which is activated when the hormone binds to the receptor. And then third is the effector protein. In the example in the book, that effector protein is the enzyme adenylyl cyclase. But there are other effector proteins out there. So part one, the receptor protein. Part two, the G protein. Part three, the effector protein. The effector protein causes production of the second messenger, which has effects in the cell. The second messenger isn't part of the receptor complex, the G protein coupled receptor. It's the result of it. Okay? So those three things that I wrote up on the board there, receptor protein, G protein, and effector protein, are the three parts of that type of receptor. And like I said, I meant to put this picture in the presentation, but I forgot to while I was eating dinner. I did, however, fix a really bad typo, so be thankful for that. Okay. Okay. As far as the presentation here is concerned, I'm going to stop the presentation there. I'm going to go a little longer. It's not quite perfect time for a break. This is when I took a break with my earlier class, which means that I'm doing better on time with you guys than I did with them. So I'm happy about that. Actually, let me leave this up to you. Either this is a nice natural place to take a break, and then we'll come back and finish things up. Or I can go on for another 15 minutes, and then we'll take a rather unnatural break in the middle of what I'm doing, and then we'll come back and finish up. Either way, we're going to go till the end of the class. Okay. It's not like taking an early break means you're going to get out early. It just means that you have a longer time from the end of break to the end of class. So which would people prefer?
Natural progression.
So you want to take a break now so that I'm not interrupting myself in the middle of things? Everybody agrees with that? Okay, so let's take a break now. Be back in here a little bit before the top of the hour would be best, just so that we have as much time as possible. So about 20 minutes, and we'll continue on.